Okay, welcome everybody. Today is a special treat made possible by serendipity. This lecture is possible because some of you might notice we get a few journals for the center, and one of them is a quarterly review of biology, and I browse it, and I came upon this book that looked kind of interesting, Biochemical Adaptations. And I thought, wow, I've always hated biochemistry because they just made me memorize things without thinking. Maybe it's interesting. Got it, opened it up, and oh my gosh, it's the book I have always wanted, and I would love for medical students to have access to it because you can actually remember things better if you have the kind of special framework in the evolutionary perspective that George Somero is how most people pronounce it, but it's Finnish, so it's Somero, or close to that.
Close to that, yeah.
So there are further serendipity and wonderful connections. He actually did his undergraduate work at Carleton College, an institution that I just love for very personal reasons, having gone there a few years after George did. He then went to Vancouver, UBC, for a postdoc. He was at Stanford. He ran the biological station at University of San Diego for a time, and then, but of course, another thing about him is that he's not an Arizona kind of person, really. He loves snow and ice and spent several years at McMurdo early in his career. The summary of his work, however, is that he is the person who's probably done more than anyone else, in recent years especially, to bring real evolutionary thinking to biochemistry. And it seems to me that this is our best opportunity to really connect phenotype with genotype, the challenge that we're always facing. He has a bunch of good examples today. I'm just going to remind you one he hasn't mentioned that you all know about probably. He's the person who discovered that when you raise the temperature of Arctic fish to 4 degrees centigrade, they die of hyperthermia. It's the kind of thinking that has caught on and made for major advances. Over to you, George. We look forward to your talk.
Okay, thank you. Well, I want to thank all of you really for coming, listening to this spiel. I really enjoy my time at Arizona State. I've done a number of talks here over my relatively long career, and I've just been hooked up with Mike to come back and spend a week and continuing the discussions that we began this morning. So what I'd like to do during the next 45, 50 minutes is to look at an aspect of molecular evolution that is not commonly on people's radar screens. And this is what I'm calling micromolecular evolution. I'll define what I mean by a micromolecule in just a moment. I'm not talking microRNAs, I'm talking really small molecules that usually evolutionary biologists don't even know exist. And what I want to show is that macromolecules, the proteins and nucleic acids, and big things like membranes that physiologists and evolutionists are primarily interested in, sustaining their function is one of the roles of the cellular soup, as I call it, in which all these big molecules live. And just to give you a perspective on what's in the cell, this is by the total number of molecules in the cell. Macromolecules, DNA, RNA, or proteins, a relatively small sliver in terms of quantities of molecules. For example, at the extreme, in some bacteria, there may be only one DNA molecule. So most of what's in the cell are what I'm calling the micromolecules. Water is 55 molar, inorganic ions, small organic molecules, and protons that I'll come to at the end of my talk. So this is what constitutes the cell. This is how funding is distributed in biochemistry. It's essentially the mirror image in terms of the number of entities versus the number of dollars. And one point in showing these two pie charts is to point out that there's been a failure to recognize how important micromolecules are in facilitating the functions of cells and contributing to the evolution of organisms in very different environments. So we really have what I'm calling narrow-mindedness in biochemistry. As Randy said, most biochemistry classes, including the one I took at Stanford, but not the one I teach at Stanford, is memorizing structures, memorizing names, memorizing pathways. And that's a very poor way to learn biochemistry. What I'd like to do is to tell you what I think are some interesting stories about differences in biochemical systems among organisms and show you how important it is to take what I'm calling an integrative approach, where we look at the little guys as well as the macromolecules, to understand both how organisms evolve over long multi-generational processes and how they acclimatize to acute changes in their environments. And in case there are any ecologists in the audience, I'm I'm sort of pandering to you, but we really have to, biochemists have to begin thinking more like ecologists. It's a complex interacting web of big and small chemicals that allows organisms to do what they do. So since I began graduate study in the 1960s, and as Randy said, I worked in Antarctica with these fish that have body temperatures of -9 Celsius, freezing point of seawater. They're ectotherms in thermal conformity with the water. I've also worked at deep sea hot springs where the organisms can have cellular temperatures, archaea, up to about 122, 125 degrees. One of the most remarkable things about life, speaking very generally, if you'll allow me to, is that we find the same types of biochemical functions and structures are present in all organisms, by and large. ATP generating abilities, abilities to transmit genetic information, to make proteins. And the macromolecules that are responsible for these common activities among organisms are highly sensitive to abiotic factors, temperature, pressure, solution conditions. They can be very disruptive of macromolecules. So how do organisms manage to colonize all these different thermal niches or hydrostatic pressure niches in the case of some of the deep sea organisms? And that's sort of the general context in which I'm going to present my stories today. The short answer to that question about how do organisms adapt to all these different abiotic conditions is that there really are two general adaptive solutions for coping with abiotic stress. One of these I call, just for convenience, intrinsic adaptations, to indicate that here we're talking about evolutionary changes in macromolecules. And I'll focus today on proteins, but as I was proselytizing a few moments ago to Mike and his group, the same thing applies in terms of nucleic acids. There won't be time to go into that today. So an intrinsic adaptation in a protein would be a change in the amino acid sequence that would give the protein the ability to work better at high pressure, high temperature, low temperature, variable temperatures, whatever. And then coupled with and complementary to these intrinsic adaptations, what I call extrinsic adaptations, to emphasize that these are changes in the medium that surrounds the macromolecules. And I'll give you several examples of this: protein stabilizing or small organic molecules, regulation of proton activity in the cell. These types of extrinsic adaptations keep macromolecules happy, as it were. So it takes two types of adaptation to allow organisms to thrive under these very different abiotic conditions we find in the outside world. So here's the menu today. I want to first introduce two concepts that are really important. And as I say, these are the bedrock for all the discussion. So it's another way of saying, pay attention now when I start to define these things. And these are the concepts of marginal stability, which is one of the unifying principles in biochemistry. It applies to proteins, lipids, nucleic acids. I'll define that in just a moment. And the other is corresponding states. Corresponding states is almost a form of homeostasis, whereas you go among organisms living under very different abiotic conditions, certain traits of their macromolecules have the same values. I'll give you examples of that. And then I want to take a very quick look at protein adaptation. I won't have time to go into the details on what amino acid substitutions mean in terms of changing structure. I'll be happy to hang on during, I guess, what's called tea time afterwards. This is not beer, but tea. Do I have that right? Anyway, I'll be happy. Beers later. I'll be happy to expand on that discussion with you during the question period during tea time, but then spend most of my time on what I'm calling micromolecular evolution. Again, keeping proteins happy through appropriate adjustments in the small molecules in the cell, osmolytes and pH. There are a number of abiotic stressors that I'll be considering. All of them have very strong effects on macromolecules, on proteins. The one I'll spend most time on is temperature, because as a general rule, temperature affects everything an organism is or does. All structures are temperature sensitive. All rates of activity are temperature sensitive. So temperature has been justifiably one of the main focal points of scientists like myself who work at the interface between physiology and the environment. Later, I'll say a few things about osmolality, hydrostatic pressure, and reactive oxygen species to try to build a general argument concerning why cells accumulate the micromolecules that they do. So, okay, here's my attempt to define what I mean by corresponding states, and I'll do it in the context of temperature. let's say we have a bunch of species, polar species, temperate species, tropical species, and we purify a protein, the same protein, the orthologous protein from these different species. And we look at its thermal stability, which we can do calorimetrically. Commonly, it's done by looking at the rate of denaturation of the protein as we hold it at high temperature, measure its loss of activity over time. And what we find, and I'll show you some data on this, is that here's adaptation temperature, the temperature at which the organisms have evolved, approximately. And this is the intrinsic stability of structure. The intrinsic stability almost invariably, I'd say invariably, goes up as we look at organisms adapted to higher temperatures. And one example, and I'll return to some of these organisms later, this is sort of looking out my window at Hopkins Marine Station, and we have a number of snails that occur at different heights in the intertidal zone. Tegulimonterei is almost always underwater, which means it's about a 12-degree snail. Some of the littorine snails that occur high up can see temperatures upwards around 40 degrees on a hot day at low tide in the summer. So we isolate a common protein. This is malate dehydrogenase. Follow its loss of activity at 42.5 degrees, and we get this series of denaturation curves. And the slope of this curve is a direct reflection of whether the organism is cold or hot adapted. So it's a very consistent pattern, not only over broad ranges of latitude, but even at a single latitude as a function of the distribution height. And this has been seen now in quite a variety of proteins, quite a variety of organisms. So it's a general pattern. Now, with corresponding states, it's kind of intuitively obvious that if you're a high body temperature organism, you want to have tougher proteins because of thermal stress on protein structure. But when you level the playing field and look at stability, here's again adaptation temperature, here's thermal stability at the normal body temperatures. The previous slide showed data that were gathered all at a 42-degree denaturation temperature. Here, when we look at organisms at their normal body temperatures, we see a similar stability that's often referred to by protein chemists as a corresponding state. So through evolution, there are differences in the intrinsic stability of organisms that evolve at different temperatures. But when you look at the same protein at the normal adaptation temperatures of the organisms, their stabilities are very much the same. Okay. And that's what we mean by corresponding states. And there follows from this corresponding states of function, which I'll show you in a few slides. So you can say, well, okay, that's the pattern, but can we rationalize it? Why does it matter for this corresponding state to exist for proteins, such that a protein in a polar fish and a tropical fish have similar stabilities, similar flexibilities at their normal operating temperatures? And we can go back, almost go back to Aristotle. I made this slide, I had the Goldilocks principle. I thought, no, I was giving a talk in China. I thought they might not understand who Goldilocks is, but surely they know Aristotle. Confucius never spoke on this topic, but Aristotle talked about the golden mean. Not too strong, not too weak, this kind of thing. Or Goldilocks, not too hot, not too cold. And the point is that when proteins evolve, they attain a structure that's not too flexible and it's not too rigid. And again, protein chemists have a term for this, marginal stability. So the proteins are stable enough to hold together, but they're not so rigid that they can't function well. And this might seem to be a paradox because on the one hand, you think, well, it's expensive to make proteins. You don't want them denaturing. You don't want to have too much energy put into protein turnover. Why not make proteins as tough as they could possibly be? Wouldn't that be advantageous? But we can resolve this paradox very easily when we look at what proteins do and how protein function is related to changes in protein conformational structure. So in other words, a protein has to be strong enough to ensure retention of the basic three-dimensional structure it needs, for example, to recognize ligands, substrates, cofactors, other proteins. Yet it has to be flexible enough so it can change its structure during function. And every enzyme that's been studied, hemoglobins are another example, when the protein is carrying out its function, it's changing its three-dimensional structure. And that's critical for all proteins. So it's kind of a balancing act. I'll show you two slides to get that point across. One is the enzyme has to be rigid enough so the geometry of this binding pocket, this is for an enzyme lactate dehydrogenase I'll say more about through the talk, It binds NADH and pyruvate, or can bind lactate and NAD, but it has to have the right geometry to allow this binding to occur. So that's important. And subsequent to binding of the substrates here in the binding pocket, the protein changes its conformation by almost 20 angstroms. It'll change conformation, the chemistry goes on, and you make products. So again, stable enough to recognize the entities that the enzyme has to work with, flexible enough to carry out its function. So there has to be a balance between rigidity and flexibility. Now, I'll be focusing in some of my presentations today, some of the stories, on the binding event, because it turns out that the geometry of the binding site is very sensitive to abiotic conditions, temperature, hydrostatic pressure, solution composition. And temperature can distort the geometry of binding sites very, 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 very readily. And that, as you'll see, is an important aspect of protein evolution to temperature. But when we look at the rigidity of binding sites, rigidity, flexibility, I'll kind of use these terms interchangeably, what we find is that binding site rigidity increases with adaptation temperature, much as the stability of the entire protein increases with higher adaptation temperature. The specific binding site shows this same sort of effect. And we have a paper we're submitting next week on this process. And I couldn't resist making a last-minute slide to show this to you. And you'll be a little bit dissatisfied because I can't give you a talk on molecular dynamics simulations that were used to gather these data. But if you take at face value that we have an in silico method for measuring binding site flexibility, which we do, which can be very high to very low, depending on the organism you're looking at. Again, these are all measurements made at a common simulation temperature. Here are the snails that we're working on in China. They're shaman. This is the intertidal zone at Shaman. This is the intertidal zone at Monterey. Remarkably different in conspicuous richness. No macroalgae here, cyanobacteria in the rocks, some microalgae. Temperature is getting up above 55 degrees. The snails we work with tolerate temperatures between 54 and 56 degrees. That's their upper lethal temperature. And as I was telling somebody, this is like a snail that could crawl along the sidewalks in Phoenix on a very hot day where somebody is frying an egg on the sidewalk. That's what we always hear happens. These snails are remarkably heat tolerant, and I don't have time to tell you more about them, except that when you look at the flexibility of binding sites of their enzymes, they're very, very high. So this is the thermal tolerance of the animals. These are 55-degree animals, very stable, rigid binding sites compared to those that we find in colder adapted species. We've done this now for 11 species, like the ones found at 12 degrees. So there's selection for overall protein stability, and there's, I think, very strong selection to keep the binding sites geometry in the right form at these very high temperatures. Because a protein that has to work at 55 degrees, you know, just intuitively, well, that's going to be a real challenge. And these snails do it really well. But that could be a whole other seminar. So corresponding states of binding through the amino acid substitutions that affect protein stability, flexibility. Here we have an index of binding. This is for lactate dehydrogenase. Lactate dehydrogenase and malate dehydrogenase are essentially the same enzyme, so I'm going to flip back and forth between them. They're sister enzymes. You can interconvert them with one or two amino acid substitutions in the active site, changing from malate to pyruvate. So LDH is a good example, one we've worked on for a long time, going from desert iguanas. And no, Randy, I am a desert person. I taught a course in Death Valley when I was back in San Diego. KM is an indicator of binding ability. What you see is that in every case, as you raise temperature, the KM goes up, and that refers to a decreased binding ability. So low KM means tighter binding. I won't go into the textbook material on that. Just take that at face value. So for every ortholog of LDH or every ortholog of MDH as well, as you raise the experimental temperature, this now is experimental temperature, KM goes up, which means binding goes down. But thanks to these changes in amino acid sequence that affect flexibility, you find that essentially the same KM is maintained across at least a 40, maybe a 45-degree range of body temperatures, from the very thermophilic desert iguana down to the Antarctic notothenioid fishes that I cut my teeth on as a grad student. So that's what I mean by corresponding states of function. Those are facilitated by corresponding states of protein flexibility. Okay. Well, how do we how do we ensure that when an organism is seeing acute changes in abiotic factors like temperature or hydrostatic pressure, as I'll show you, that all the work of evolution isn't for naught, that the protein can continue working well. And that's where we get into micromolecular adaptations. And I'm going to close talking about pH. Most students hate pH. It's not just that it's a negative logarithm, which is hardly intuitive, but they don't see the importance of it. I'll try to show you why pH is so important. I first want to look at osmolytes, the small solutes that are numerically dominant in the cell, as you saw on the first pie chart I showed. And when we do a survey of marine animals, and I'll flesh out some of the details in this slide. In teleos fishes, they're much less concentrated than seawater. Here's seawater, mostly sodium chloride with sulfate and magnesium, several millimolar concentrations. Marine invertebrates have very high levels of free amino acids, FAA. Sharks and their relatives have a lot of urea and glycine, betaine, and trimethylamine-N-Oxide. So we see a variety of organic molecules being used to build up in these organisms that are isoosmotic with seawater. In other words, the same osmotic concentration as seawater. It seems to be a game that you can play with different types of resources. We see strong conservation in some cases, particularly in the case of potassium ion in the cell. And when you look at the effects of potassium ion on proteins, for instance, they can be very perturbing. So if you move too far away from the canonical concentration of potassium, maybe 125 millimoles, it's perturbing. So the osmotically concentrated organisms don't vary the inorganic ions, they vary organic osmolytes, and we'll see why that's the case. So there's this oddball mix, and when we first began this work many years ago, there were no rationales, no explanations for why marine organisms use this odd variety of small organic molecules. And what I want to convince you of is that there are some bases for explaining why certain types are used, why certain types aren't used. And the conjecture, the hypothesis I'll be examining is that by designing the right, I shouldn't use that word, by establishing the right type of organic soup in the cell, we can preserve the marginal stability of proteins and corresponding states of structure and function. So that's kind of the simple take-home message I hope I get across. There aren't that many different categories of organic molecules. And I show this slide just to give you sort of the set of options that cells have to use in designing their organic osmolyte pools. Carbohydrates, trehalose is very important. John Harrison, others who work on insects know that story. I won't get into that story. Amino acids and their derivatives, including taurine, beta amino acids, glycine, proline, are commonly used in marine invertebrates and marine bacteria. In some ways, the most interesting soup that I'll be talking about is the one that's found in chondrichthyan fishes, shark, skates, rays, and the coelacanth, some of you are familiar with. And these are methylammonium and methylsulfonium compounds, like trimethylamine-N-Oxide. They're usually paired, not invariably, as you'll see, they're usually paired with high levels of urea, levels of urea that are high enough to be fatal to ourselves. So one question is, how do sharks get by with this? So let me give you two key facts about the effects of organic osmolytes. One is that they're not all the same. You go from one osmolyte to another. And urea is a good example. Urea is a very strong denaturant. Biochemists typically use it to denature proteins. Other organic molecules are very strong stabilizers, and their stabilizing power varies a lot from chemical to chemical. I'll show you examples of that. So one thing that cells will do is to strike the right balance between stabilizing and destabilizing effects. You don't want too high a stability because that would be detrimental to the flexibility of the protein. You don't want the protein to be too flexible because then it's not going to be able to have the right three-dimensional structure. Another fact is that a given organic osmolyte tends to affect all proteins similarly. So there's a potential for a global solution across the entire proteome. If you need to build up an osmolyte to achieve some goal, let's say dealing with pressure or temperature, you can change the right osmolyte and all the proteins in the proteome will be affected similarly, by and large. And that's an important point. It makes life easier when you're acclimatizing to change in an environmental factor that perturbs proteins. So what I want to look at then, I've already sort of stated this hypothesis, is that we're looking for these global solutions for dealing with, in this case, with thermal stress, looking to see if stabilizing osmolytes build up when organisms are given increasing temperatures, and do they come down in concentration when the system is at cooler temperatures. And I'm going to look initially at the rocky intertidal zone outside of my window at Hopkins Marine Station. So it's a very temperate environment, but it gets a very wide range of temperatures. I mentioned earlier, as you go high up on the rocks to low down the rocks, you see a very large difference in temperature. And these are field temperature data recorded for mussels on the rocks. Over the season, a change in temperature of 30 degrees Celsius. During the course of a day, temperatures can vary 15 degrees, let's say, depending on how clear the atmosphere is, what's the tidal cycle. Anyway, it's a system where a given type of ectothermic organism, cold-blooded organism, is going to see a wide range of temperatures daily with the tidal cycle, annually with the seasons. And we put this local ecosystem to good use by looking at different sites, tide pool sites, high sites where the organisms get strong sun exposure. This is a southwest-facing site that gets really warm. We've recorded temperatures up to 37, 38 degrees in the animals, the mussels on the rocks. And low sites, where they're shaded, facing to the northeast, they don't see much sunshine. What we found was that when we measured the levels of osmolytes, organic osmolytes, in these organisms, glycine betaine, a methylammonium solute, a good stabilizer, increased with temperature. Perhaps most interesting was taurine, which is both a protein stabilizer and an antioxidant. So the organisms that were experiencing higher temperatures, we were measuring their body temperatures in the field, so the temperature data are reflective of the actual conditions the animals saw over time. In the field, taurine levels built up as temperature built up. And taurine, shown here, is not only a protein stabilizer, there's some evidence that it stabilizes membranes, which is another important part of the picture, but it's also an antioxidant. So just to summarize, again, you have to take a holistic perspective on looking to see how organisms acclimatize to environmental change. You want to conserve the properties of their proteins, and these stabilizers like glycine, betaine, and taurine can maintain a corresponding state of structure that balance between the golden mean balance between flexibility and rigidity. Some of these have antioxidant properties, so as metabolic rate goes up at higher temperature, you have a higher capacity for coping with reactive oxygen species, and of course, osmotic balance is maintained. Other solutes, for example, glycine, go down as taurine and glycine betaine go up. So that's a very quick trip through the intertidal zone. And I want to go offshore a little ways and look at another group of organisms that's long intrigued us. These are marine chondrichthyans. What are they up to? Because invertebrates vary free amino acids. They change the types of free amino acids they have. They change glycine, betaine. But sharks and their relatives, this is a blue shark, and this also is in rays and coelacanths, they have this funny mixture of urea and trimethylamine-N-Oxide. And that raises two questions. First of all, why this mixture? Why don't they just use free amino acids the way invertebrates do? Why do they use TMAO, glycine betaine, and urea? And why the concentration ratio that we see? Because consistently, when you're looking at shallow living chondrichthyans, and I'll get to the deep ones in just a moment, you find that it's about a 2-to-1 ratio, twice as much urea as TMAO. And we pondered that. And Paul Yancey, graduate student in my lab, worked out what's going on here. And I think it was a very important discovery because it was one of the first examples of how these complicated soups of organic osmolytes represents a balancing act between stabilizers with different potency. TMAO is a much more potent stabilizer than glycine betaine, and a destabilizer like urea. And I'll show you two slides taken from the book that Randy mentioned, but taken initially from Paul Yancey's thesis work. What he showed is that when we're looking here at the binding ability of an enzyme for its substrate, this is a pyruvate kinase. We're looking at the binding of adenosine diphosphate. Again, Km is our criterion. Urea and TMAO have opposite effects. As we add urea the Km goes up. As we saw with high temperature, the binding ability is falling off as you increase the urea concentration. That's almost invariably seen because urea is distorting the protein's three-dimensional conformation. You put trimethylamine-N-Oxide in on its own, and the Km goes down. The binding becomes very tight. But protein flexibility maybe is too extreme. And what you see when you look at the physiological mixture that's found in sharks, with urea being at twice the concentration, you can see from the labels on the two abscisses, you're right back to the control value. So urea pulls a Km up, Km pulls it down, put urea and TMAO at a 2-to-1 ratio, and you're back to zero, basically. So it's a counteraction effect, or algebraic additivity. A plus and a minus cancel out. The same thing is true in protein structural stability. And my favorite data set from Paul's work as a graduate student is shown in this lower frame. What we're looking at is a change in absorbance of a protein as we monitor its unfolding during incubation times out to about an hour. And what this reflects is the labeling of sulfhydryl groups. This is a glutamate dehydrogenase preparation. You add urea and you see the labeling occurs faster. That means the protein is more flexible. It's alternating through conformations that gives this labeling tag with this absorbance here around 420. That attaches to the protein much more quickly when the protein has a loose structure. TMAO slows down the rate of labeling, makes the protein tight. Control value and urea plus TMAO basically the same curve. So again, it's this balancing act, this counteraction that explains why organisms like sharks use TMAO and urea in this 1-to-2 ratio. So the question is, well, this is pretty neat. I mean, and if something is discovered in one evolutionary lineage, it's quite often discovered in other lineages if we know where to look. One place to look, and this discovery came apart developed based on the work that had been done with sharks, and that's in the inner medulla of the mammalian kidney, where urea concentrations can get very high, especially in organisms like kangaroo rats, where urea can be over 1 molar in concentration. And for many years in the kidney literature, there was something called the osmotic gap. People knew there was a lot of urea, but there were other organics which hadn't been characterized. And it turns out that what fills the osmotic gap are two types of compounds. These are two sugars I'll mention, but the thing that's, I think, most interesting is glycerophosphoryl-choline, a compound that had been detected, but nobody quite knew what it was for, what it was, it's just another waste product that the kidney is getting rid of. Well, glycerophosphoryl-choline is a fascinating molecule because it's like a designer stabilizer. Or a super stabilizer, as I called it here, because it has 3 stabilizing groups knitted together in one molecule. We've got a methylammonium tip, like trimethylamine-N-Oxide or glycine betaine, so that's going to stabilize. Phosphate ion is a very strong protein stabilizer. Glycerol is also a protein stabilizer. So what happens when an organism gets a high protein diet, maybe doesn't have a great deal of water, urea concentrations build up in the kidney, especially in the inner medulla, is that glycerophosphoryl-choline increases in parallel with urea concentration. Sorbitol and inositol also build up, but glycerophosphoryl-choline is a remarkably strong protein stabilizer. We think this is what the mammalian kidney is using to offset the effects of urea and keep cellular functioning going at a good level, even at urea concentrations of over a molar concentration, as you see in some of the desert rodents. Well, that's temperature. Let's look at another physical factor. We've seen how temperature effects could lead to acclimatization using these different solutes. What about hydrostatic pressure? And just kind of a basic primer for those of you who aren't oceanographers. As we go down in the marine water column, with every 10-meter increase in depth, pressure goes up by 1 atmosphere. So pressures can get up to about 1,100 atmospheres in the deepest parts of the ocean. Surprisingly, again, going back to my fourth slide, all the same kind of biochemical processes go on at pressures of 1,100 atmospheres. Something has allowed that. A lot of organisms, invertebrates, certainly bacteria and archaea, can live at these 1100 atmospheres pressures. Fishes, both bony fishes and cartilaginous fishes, are limited to 8 kilometers. And I'll give you, Paul Yancey, again, has done some beautiful work on this to explain why fishes kind of hit a brick wall around 8 kilometers depth. Pressure has many effects on proteins. It can distort their shape, it can disrupt enzyme binding, it can denature them. It also compresses the volume of the cell, and I'll allude to how solutes help get around that problem too. Again, we can ask, as we do with temperature, is there a global solution to dealing with the effects of high pressure? Not only for organisms that live at a constant great depth, but many marine organisms migrate through the marine water column over very wide pressure ranges of hundreds of atmospheres. Is there some way that the organism can become uribaric as well as some organisms have become eurythermal, widely tolerant of pressures? And here's Paul Yancey, who also solved this mystery. He solved the mystery of the shark osmotic story, solved the mystery of protein stabilizing forces at depth. This is a sign he made for our lab, still hangs there. Again, here's trimethylamine-N-Oxide, a methylammonium solute. Strong protein stabilizer that's used in sharks. It also has the effect of expanding the volume of water. It has a positive partial mole volume. So when you put TMAO into say into a graduated cylinder, the fluid level rises because TMAO expands water structure. And I'll come back to why that's important. Here's a data set that's almost too beautiful to be true. Paul sampled fishes collected at different depths down to 8 kilometers. Again, this is the deepest occurring fish. The Japanese, a week ago, went back and extended the depth range like 20 meters or something and got a lot of hoopla on the press on that. But here's Paul's graph looking at trimethylamine-N-Oxide millimoles per kilogram as a function of depth. Now, most of these points represent different species, but some of the points represent a single species. I can't say which points are which, but this might be a species that was collected around 1 kilometer down and also around 5 kilometers down. So this is the trimethylamine patterning is something that shows acclimatization. As an organism goes deeper in the water column, it builds up trimethylamine-N-Oxide. Comes up shallower, it reduces the concentration. And just to kind of give you a sidebar story, Paul's argument in this paper that he published in PNAS is that when you get above 8 kilometers depth, for a fish to continue adapting to higher and higher pressures, a bony fish, it would have to become hyperosmotic to seawater, and bony fishes just don't do that. So Paul thinks it's a limitation in this osmotic pressure counteracting strategy that sets the depth limits of fishes. Just one example, again, going back to the corresponding states perspective, look at controls, 1 atmosphere controls, no added solutes, no TMAO, glycine, betaine, et cetera. When you increase pressure, binding is reduced. This is now, again, is LDH. The Km goes up significantly. That means less binding. When you put trimethylamine-N-Oxide in, that 1 atmosphere, the Km is actually significantly reduced. You put in 250 atmospheres pressure, it's back on the control value. So again, we're keeping a corresponding state of binding ability in the face of changes in pressure, and TMAO is doing that. It's more effective than the other solutes that have been examined. Threw this slide in because I mentioned earlier that when we examine shallow living cartilaginous fishes like sharks and rays and skates, it's a 2-to-1 ratio of urea to trimethylamine-N-Oxide. But urea, like hydrostatic pressure, tends to perturb protein structures. So what Paul Yancey found is that this magic ratio of urea to TMAO changes with depth. And you get down to some of the deepest collected fish, and you find that there can actually be more TMAO than urea. So there's this balancing act dealing with both keeping the osmotic conditions correct for the cartilaginous fishes, osmotic informers, but also dealing with the effects of hydrostatic pressure. So I think it's a really neat story. And it's a good example of a global solution to a problem. One molecule with the right concentration adjustments can solve a problem that exists all the way through the marine water column. So in closing section, I want to look at protons. As I said earlier, pH is something that most people cringe at. And in my teaching experience, very few people have a good sense of how to answer these two questions. Why are biological pH values what they are? And why does pH vary as it does with temperature? This curve extrapolated out using the neutral pH of water, which I won't say anything more about, because the important thing is this pK value for imidazole, which I'll spend a bit of time on. You extrapolate the thermal dependence of that, what you find is, as you get out to some of the most thermophilic archaea, their intracellular pHs are probably going to be down 5.7, 5.8, which from the perspective of someone, let's say in biomedical science, whoa, that's really acidotic. But what you're going to see is that it's not at all. That's what you want to have to maintain the cell's soup in the right condition for proteins. So blood is usually alkaline in animals. Now blood is usually alkaline to the cytosol. PHI is what we use for the cytosol. Well, what are these values about, and why this change in pH with temperature? Well, the starting point for the analysis, and I think it's the right starting point, is that proteins need a side change that can be titrated. For example, during exercise, as you're building up protons, you want to be able to activate processes that are going to take those proteins out of the system. You're going to want to change metabolic flux. And to do that, you have to have a protein that's sensitive, protein that has an antenna that can read pH. To say that a protein can be titrated is really saying that the pK of a side chain, one or more side chains, should be about the same as the intracellular pH. That makes the protein maximally sensitive to change in pH. And I'll give you an example with LDH again to show what this means. So the titration can do a number of things. It can optimize ligand binding, KM conservation, corresponding states, again, I'll show you that. And it can affect protein structure. Protein structures, particularly subunit assemblies, are often very pH sensitive. So you want to have a handle on it. You want to be able to titrate side chains to make sure that the protein is binding effectively and assembling effectively. There's only one candidate amino acid of the 20 or so that we find in cells, and that's histidine. That has an imidazole side chain. And what pH values in the cell reflect is that they're maintained in a range, in the cell now, they're maintained in a range right around the pK of imidazole side chains. For how many of you is this new? Because I worry sometimes about, a few people at least, yeah, that everybody knows what I'm talking about. But then I find, no, people haven't heard about this kind of thing before. Because temperature-dependent pH is something that biochemists typically neglect and often generate some bizarre artifacts in the process. So anyway, let's now switch over and examine why pH varies as it does with temperature in both the extracellular and the intracellular space. And the answer is that pH of the cells in the blood falls by about 0.018 pH units per degree rise in temperature. So that means that an Antarctic fish down here looks very alkaline to what it should be, but it's not. That's just the way things work. A mammal at 37 degrees from the perspective of an Antarctic fish will look pretty acidotic. But again, that's the pH that's best for that temperature. So pH changes this way identically to the change in the pK of this histidine imidazole side chain. What that means is that the dissociation state of the imidazole side chains remains stable as temperature and pH co-vary. So that gives the organism proteins that have a consistent charge state and a consistent ability to respond to changes in pH. Because again, the pK is changing with temperature along the same line as pH is changing. And this is called, R. Blake Reeves and Herman Rahn out of Buffalo, New York, developed the alpha stat concept. And alpha is simply the fractional dissociation state of imidazole side chains. So as you increase the temperature, protons tend to leave the histidine imidazole ring. So you really need a lower pH back pressure to keep the dissociation state constant. And that's what we see with alpha stat regulation. Okay. Now, in principle, there could be two patterns. Alpha stat, the temperature varying pH, or pH stat, where pH is constant over temperature. You can go, well, show me something that's going to say that alpha stat is better. So again, we go back to LDH, which has been kind of our target enzyme for many, many years. And it's a good enzyme for looking at pH relationships because the imidazole ring right here on histidine 193 sometimes histidine 195. Anyway, it's a key player in catalysis because for pyruvate to bind to the active site, there has to be a proton on this imidazole ring. So if you're increasing temperature and not decreasing pH, increasingly it's going to be hard for pyruvate to bind because that proton is not going to be there. So Km should shoot up as temperature rises, both from disruption of the geometry of the active site, but also because there's less and less probability of there being a proton on this imidazole ring. So that's why alpha-stat regulation is good. And remember this pattern I showed earlier, this very strong conservation, corresponding states of binding abilities. This you only see under alpha-stat regulation, where pH is varying with temperature. And if you're doing enzyme assays at different experimental temperatures, here from 0 to almost 50 degrees, you use an imidazole buffer because it changes pH instantly as you change temperature. And that may sound bad, oh, things are out of control, but it's actually, that's biologically realistic. And when you look at many of the papers done with a single pH value, phosphate buffer has a very minimal change in pK with temperature. You look at studies where people have looked at the effects of temperature on organisms at a common pH, or on enzymes at common pH, and you see artifacts. The reality is that you have to use alpha-stat conditions. And I'll give you one example of this in closing. Here again, these are similar data to what are shown here, just fewer species. The, the open symbols represent the pH stat situation, where you use a single pH value across all measurement temperatures. The fill symbols are doing the same experiment, but using a buffer that changes its pH with temperature, following the same alpha-stat pattern that we see in real life, basically. So here's the range of Kms that you see under alpha-stat regulation, which is what occurs in organisms. Here's the range you see under pH stat conditions that are commonly employed by naive biochemists. So this little proton is a really important player in setting up the right kind of conditions in the cellular solution. And you still see a lot of papers being done where the pH conditions are so different from what's going on in the cell that you kind of question what can you say about the data. Just a final example of how nifty micromolecular evolution is, and this again builds on what I was saying about the role of histidine imidazole side chains. Evolution has come up with some very interesting buffering compounds and intracellular buffering capacities. We often think, oh, bicarbonate and phosphate. When you look at organisms that have a very high intracellular buffering ability, this is based on dipeptide buffers that contain histidine. So they're very good buffers because the pK of the buffer is right there at the pK of the cell. That's what you want with a buffer. And the pK changes with temperature, as does the pH of the cell. So the buffering capacity is maintained across a wide range of temperatures. And you also find that the concentrations of these dipeptide buffers are directly proportional to the capacity for acid generation. In locomotory tissues, locomotory muscles that have a very high activity for anaerobic glycolysis, you see booming levels of these dipeptide buffers. So again, it's a nice example of what molecular evolution at the micromolecular level has come up with. So let me summarize, last couple of slides. So what goes on in micromolecular evolution? Well, first of all, there's strong selection for coming up with the right kinds of osmolytes, both for osmotic balance as well as for controlling pH values. We see superstabilizers like Glycerophosphoryl-choline. I didn't get into thermophilic archaea, but they have some remarkably strong stabilizers like GPC that allow their proteins to work at conditions of over 120 degrees. And then there must be selection through regulatory mechanisms, and this allows the altering of osmolyte composition, both the types and the amounts, the relative amounts, and altering pH values. We don't know as much as I think we should know about these regulatory mechanisms. We know a lot about pH, but how the osmolytes are regulated, how the muscles in the intertidal zone say, it's getting warm today, I'm going to build up glycine, betaine, and taurine, that we don't know. So I think that there's some interesting work that lies ahead on that. So final take-home message, it's kind of a buzzword, but I think it's an important one in this context. Holistic or integrative. If we're interested in how organisms evolve and how they acclimatize, we have to give micromolecules and macromolecules due consideration. We can't just sequence genes and say, oh, I understand evolution. There's a lot more going on. The majority of entities in the cell are these little things, the colored dots, not just these big globular and stringy things, the proteins. And we simply have to take this holistic approach to understand how organisms work. With that, I will thank my colleagues, Linda Holland, who I've worked with over many, many years, first at Scripps, Peter Fields, postdoc, done a lot of work in LDH, my Chinese collaborators who working with now on these 55-degree snails, which we're going to transplant to Phoenix to see if they can actually walk around on the pavement in the summer. Brett Lockwood, a recent student, Paul Yancey I've mentioned, and Margaret McFall-Nye. So I'll stop there with my remaining vocal cords. I'll be happy to entertain any questions. Thank you.
Anyone have any questions?
So with the sharks, you show the difference in So with the sharks, you show a difference in the ratio of denaturants versus stabilizers. You go down
Right.
in sea level. Mm-hmm. Does that mean that the sharks that live at lower depths have not evolved proteins to be in the proper stability at that pressure?
No, I don't think that follows. I think first of all, sharks only go down about 5 to 6 kilometers. They're limited in their depth of occurrence. No, I think, this is a bit simplistic, but a protein that, let's say, evolved in shallow fishes would work, in situ in the organism if you pumped up enough stabilizers to offset the effects of pressure. Pressure, again, this is very qualitative, but pressure is not as nasty to proteins as temperature is. It does distort them. It does affect binding values, as you saw. But you can, I'm guessing there's much less evolutionary change in proteins to cope with pressure. And we don't know much about that. That's terra incognito compared with temperature, because this one solute seems to be able to solve that trick. And I didn't mention this, but invertebrates do the same thing. They build up TMAO. We don't know what microbes are doing. They may be doing the same sort of thing with one or maybe just one or a small number of these protein stabilizers. So, yeah.
George, would you imagine that an easy way to adapt your heat tolerance in a rapid climate change like we have going on now would be to have mutations in proteins that regulate the stabilizers, these microcellular contents?
Absolutely,
because
yeah.
then you get all the other proteins that you wouldn't need mutations in, right, stabilized at once, is how I understand your model.
Mm-hmm. Well, people often refer to acclimatization or acclimation. The words, unfortunately, are kind of used interchangeably when they shouldn't be. But anyway, acclimatizing to global change, temperature conditions particularly. A short-term solution to the problem, no pun intended, well, pun intended, is to change the solution in which the proteins are working. That may buy you a few degrees. It's not going to buy you 10 degrees, but it may buy you enough to get through, say, a couple of centuries of change. But that's all. Eventually, as we were discussing earlier this morning, a change in average environmental temperature for an ectotherm of 2 to 3, 4 degrees is plenty to kick natural selection into action and change the amino acid sequence. And I didn't have time in this talk. I took out the slides that show how amino acid substitutions do this, but you can change one amino acid substitution in these 333 residue MDHs, and that's enough to change. Let's say you add 3 hydrogen bonds when you add a serine for a glycine. So you don't have to do a wholesale reengineering of the proteins. A single amino acid substitution very commonly is enough to change the thermal properties. So how quickly can this go on? Well, it's going to vary from protein to protein, organism to organism, population size, so on. In the meantime, these stabilizing osmolytes can come in and handle that job until the proteins are just too stressed out.
And do you think it requires few mutations to tweak these levels of osmolytes, like one regulatory protein, or we don't know yet?
We don't know yet. See, one interesting thing about some of these osmolytes, and urea is a good example. They're so-called waste products. They're protein breakdown products. So degradation of proteins, for example, might create a pool of these stabilizing osmolytes or destabilizing osmolytes, depending on what one you're looking at. So that could be one regulatory handle, but we just don't know enough about how temperature affects these or how pressure controls these TMAO levels with depth.
So George, if I understood it right, you were talking in part about a facultative adaptation where the concentration of these osmolytes is changed within an individual as they go in different circumstances. What do we know about their ability to detect the temperature and what mechanisms are mediating the changes in the osmolytes?
There are a variety of hypotheses about temperature detecting mechanisms, and there are some well-established mechanisms, particularly in warm-blooded vertebrates. I was telling some of you about, how many of you, I got to ask this question, how many of you know about RNA thermometers? Great, because I'm writing a short commentary for the JEB to bring these to the attention of non-bacteriologists, put it that way. These are prevalent bacteria as a temperature sensor. They're RNAs that have things like these stem structures, hairpin structures that melt under slight changes in temperature. These RNA thermometers have a temperature sensitivity where they can go from off to on over 1 degree change. And where they're found, the most interesting case in some ways is in pathogenic bacteria. You go out for a jog and there's dust in the air and there are pathogenic bacteria floating around, kind of metabolically inactive. You inhale them, you get them in your sinuses, suddenly the bacterium is at 37 degrees. Pre-existing messenger RNAs melt in some regions so they can then bind to the ribosome and translate the pathogenic products. Bacteria are just nasty suckers.
If we could disrupt that, that would be pretty cool.
Yeah, well, it's one example of temperature sensing. And as I'm going to say in this article, I hope to submit next week, you've got what's called translation on demand. The message is pre-existing. There's constitutive transcription of the genes for these pathogenic proteins. But there's no point for the bacterium to crank out these proteins until it's found a target host. So when it goes into your nose or down your tummy, temperatures get to 37 degrees, the RNA melts, and that puts it into a three-dimensional configuration that allows it to be translated. So it's a lot like what I've been saying about proteins. It's a marginally stable RNA, and you warm it up slightly, changes its conformation, can be translated, and then its pathogenic properties go on. That same mechanism is true for heat shock proteins. Some heat shock proteins in bacteria. The gene for the heat shock protein is synthesized constitutively, but it's not translated until temperature goes up to the point where it melts some of these small RNA structures in the 3' end of the RNA. Then they can bind to the ribosome and translation takes off. So that's temperature sensitivity. I mean, that to me is the best established mechanistic thing for temperature sensitivity.
Hi. I had a question about the regulation through the evolutionary stages of the genetics of these micro molecules that you're talking about, because I'm kind of curious if you think that it's mostly a passive response that the organisms are dealing with regulating the macromolecules with the micromolecule components like the osmolytes, or do you think there's actually a very strong genetic aspect that's regulating the micromolecules just as much as adaptations in the amino acids of macromolecules for adaptation?
Yeah, I said earlier that a lot of these concentrated osmolytes are actually degradation products, breakdown products, often called waste products, urea, again, being a good example, or trimethylamine-N-Oxide waste products. I don't know if there's kind of like a master controller somewhere where it says, oops, it's getting warm, let's turn on production of these protein stabilizers. That may be known, but I don't think it is. And if it's a waste product, there may be some way of increasing the turnover, let's say, of proteins to produce free amino acids. There's some evidence that with invertebrates that are very urihaline, they can go from full-strength seawater into estuarine conditions, and they vary their free amino acid levels in accord with the osmotic differences they see. They can increase protein degradation to free up amino acids to use for osmotic purposes. So there's some control on protein degradation. The same thing could be true in generating methylammonium solutes, but I don't know. I don't think anybody has really nailed that down.
Last question.
Yeah, I'm kind of curious as to how this compares with macroevolution in terms of, say, you have two different species from two very different evolutionary lineages, but they have to deal with the same abiotic stresses, say deep diving. Like, let's compare emperor penguins and sperm whales. They both have to dive to deep depths. Do you think that the micromolecules in both these species have adapted to in the same way? Do they have the same chemicals, or do they have different chemicals that serve the same functions?
Yeah, I can't speak to birds or mammals on this because, to my knowledge, no one has looked to see if deep diving mammals, sperm whales probably being, or elephant seals being a good example, a couple kilometers up and down too, which is another challenge. Do they have pressure offsetting solutes? That has not been looked at. But there's very interesting convergent evolution in the use of TMAO. I mentioned you see it in elazobranchs, you see it in bony fish, and you see it in invertebrates. So the same molecule is being utilized by very different taxa, long separated in their evolutionary histories to solve a common problem. You see this, I didn't talk about carbohydrates, but you see this with trehalose and glycerol. They're multifunctional, multitasking small molecules. They're membrane stabilizers, protein stabilizers, osmotic agents. So they pop up, glycerol, for example, you see it in yeast and insects in terms of dealing with temperature. So it's a molecule that performs many, many functions in essentially all organisms, but its regulation with respect to changes in temperature is very precise depending on conditions. Freezing resistance versus heat resistance, glycerol can come in both cases. So anyway, we've turned 1 o'clock and Randy's looking at me, so I better shut up. But I'll be happy to talk with you after this.
Thank you so much, George. There's tea time and further conversation next door right now for those who are interested. Thanks so much, George.